Nicholas obtained his PhD from UC Merced. And before joining AgBiome, he was a postdoc at Oak Ridge National Laboratory, where he worked primarily as part of the plant microbe interface scientific focus area. The title of his talk today is Ecological and Genomic Responses of Soil Microbiomes to High Severity Wildfire, very topical, linking community assembly to functional potential. Thank you.
Thank you. Good morning, everyone. My name is Nicholas Dove, and like Tanya said, I'm a microbiologist at a company called AgBiome. But today I'm going to present on some of my work that I did during my PhD at UC Merced and my postdoc at Oak Ridge National Laboratory. But before I begin, I'd like to briefly acknowledge my co-authors, Stephen Hart and Neslihan Tosh, and also acknowledge the funding sources, especially JGI's community science program. I had a small scale grant awarded during my PhD. Sorry. During my PhD, and really positively influenced my whole career trajectory. So really grateful towards that. So on the news, fire is generally portrayed as a devastating catastrophe. And in many ways it is in terms of its impacts on human life and property. So I don't want to minimize this. But in many ways, fire is also a fundamental ecological process. And as you can see, at any given point in time, millions of hectares on Earth's surface are burning, which results in distinct fire-adapted ecosystems, such that a world without fire would be drastically different. However, even though fire is essential in structuring many of Earth's ecosystems, it's important to recognize that fire regimes, or the ways in which fires burn, are changing. So for a variety of reasons, many of which are human-linked, fires are increasing in size, severity, and frequency, which is an issue because ecosystems that have been adapted to certain types of fires are becoming less and less adapted. And unfortunately, this threatens the sustainability of many ecosystems, prompting even the UN's environmental program to ask the question, are these megafires the new normal? So these are examples of what ecologists call state changes, where a disturbance or multiple disturbances are so severe that a ecosystem, instead of reverting back to its previous state, becomes a new ecosystem altogether. And this is generally considered to be a negative consequence. And so as a microbiologist, I'm interested in how microorganisms might mediate these different recovery trajectories. So for the past few years, I and others have been trying to answer questions related to the impacts of fire on microbiomes, ranging from basic to applied. And so we want to know, how does fire impact these microbiomes? How long does this impact last? How then do these microbiomes influence these different recovery trajectories? And then finally, can we leverage soil microbiomes to jumpstart recovery towards desirable outcomes, away from some of those state changes that I showed earlier? So to determine how long this impact lasts and what the impacts are, we use the Sierra Nevada fire chronosequence. And this is a space for time substitution technique where we go to different fires of different ages and we sample those soils and sequence them to look at how the microbial communities recover over time after what would be considered a single fire. And we use this strong location selection criteria to really minimize location impacts so that we're really isolating the effect of fire. And so, interestingly, we found the effect of fire is long lasting. So even though the disturbance is over in a matter of days, its legacy can last for decades. And so we find that at different times in this recovery succession that we have these distinct microbial communities. Taxonomically, this is a shift from a microbial community that is abundant in actinobacteria towards one that becomes more abundant in proteobacteria and acidobacteria. A key question is what is causing these differences? And for this, we looked at different assembly mechanisms. Now, ecological assembly can broadly be characterized as stochastic assembly, and this is ecological drift and priority effects, and deterministic selection, so these selective processes that result in homogeneous microbial communities or these more variable ones. And so using a null modeling technique, the beta NTI, or the phylogenetic difference between the null community and the observed community, increased in its divergence from 0 with time since fire. And what this means is that there is greater homologous selection as you move away from the last fire. And so the selection we found was partially caused by changes in the soil chemistry, particularly when we look at soil carbon compared to the four-year site, compared to our late successional site. The late successional site was about double that of the four-year site. So that's really underscores the severity of these fires. And then to a smaller degree, we found that the vegetative cover type also played a large role in structuring these communities. Now, to understand what is happening here at a higher biological resolution, we assembled, we binned metagenome assembled genomes, or MAGs, and these are these biologically distinct units. And we characterize them based on their normalized abundance with time since fire as either early successional or late successional. And what we found was that this successional classification was taxonomically resolved similar to our amplicon data, whereas early successional MAGs were far more likely to be actinobacteria, and these late successional MAGs were far more likely to be proteobacteria or acidobacteria. And since my talk is the K-base user talk, I'll also point out that the MAGs and the taxonomic and functional annotation of them can be found on a K-base at this URL, on this narrative. I don't expect people to frantically write these down if they're interested in it, so come see me after, or message me on the platform for more information on it. So to understand why certain MAGs occur as either early successional or late successional, it's important to understand what is the post-fire condition. And so first of all, post-fire soils are enriched in ash or biochar or pyrogenic carbon. I kind of use all of these terms similarly. And these carbon structures are enriched in these polyaromatic compounds that are really difficult for most microbes to degrade. And frankly, many just do not possess the enzymes necessary for this degradation. So if you're a microbe with the ability to then degrade this carbon source, you have an additional carbon source available to you, and you're likely apt to succeed. Similarly, post-fire soils are really stressful in terms of solar radiation and water stress, because this organic layer that is originally protecting the topsoil has since been combusted. So if you're a microbe with strong stress tolerance, you're also more likely to succeed. And finally, we know that post-fire soils are depleted in microbes. And so if you have fast growth rates and you're able to get a foot in the door, able to capitalize on priority effects, you're also likely to succeed. So our hypothesis here is that early successional microbes can degrade carbon, pyrogenic carbon, are stress tolerant, and are fast growers. Now, starting with the ability to degrade pyrogenic carbon, we found that early successional microbes were far more likely to possess the genes encoding for enzymes in these aromatic carbon degradation pathways. And so what I'm showing here is the normalized presence, and these warmer colors indicate that these early successional microbes are far more likely to possess these enzymes. We also found that early successional microbes are far more likely to possess the genes encoding for stress tolerance. So ectoine and mycothiol are important for osmotic stress responses, and the sigma B stress response is a pretty cosmopolitan stress response system encoding for radiative stress, heat, water, and salt stress. And then finally, we used GRID to measure in situ microbial growth rates. And so this is a peak to trough coverage ratio that estimates microbial growth rates at different points, time since fire. And so our hypothesis here was this is actually in contradiction to our hypothesis, which we expected that early successional microbes would be faster growers. And we actually found that these mid-successional microbes were had the fastest growth rates. And upon reconsideration, this actually makes sense. At this point in time, we have more carbon, in the soil, more nitrogen in the soil, more resources for these microbes to grow more quickly. And so to understand how these changes in the microbiome influence ecosystem recovery, we also looked at the functional genes in our metagenomes. And so here I'm presenting differences in carbon degradation potential using the KZ database. And so directly after fire, we see an increase in KZ abundance, especially those associated with cellulose degradation. And as time since fire increases, we see genes with lignolenic degradation potentials increasing. And so this sort of shows this sort of suggests a changing microbial metabolism with time since fire, which might impact carbon storage on these landscapes. We also saw an enrichment of nitrogen cycling genes directly after fire, particularly that nitrogen fixation and ammonia oxidation, two important nitrogen cycling pathways. And we find that this overall correlates to the inorganic nitrogen flux also measured at these sites. And so we see that there is this strong control of microbial communities on key resources. We also found that fire negatively impacts both the diversity and colonization of mycorrhizal fungi. As we saw from the last talk, that's very important. Here I'm talking more about ectomycorrhizal fungi as opposed to arbuscular mycorrhizal fungi. But we see that mycorrhizal diversity sort of recovers to its previous state around 1 to 2 decades post-fire. And this was actually strikingly similar to a recent meta-analysis that we conducted that also shows that mycorrhizal colonization recovers at around the same time period. So as mentioned, this research is ongoing. So we haven't yet begun on some of the more applied science, but with my time remaining, I'd like to talk about what could be done and what some of these desirable outcomes might be. And so the hope is that with a greater understanding of post-fire microbial assembly patterns, we can design microbial inoculation strategies that would increase plant sustainability, forest productivity, water quality, and carbon sequestration, because as we know in this room, microbes play a large role in all of these. So in summary, we find that fire selects for taxa with the ability to degrade pyrogenic carbon and survive stressful post-fire conditions, and that this impact of fire is long-lasting, can last for decades. And then finally, we find that fire can negatively impact mycorrhizal colonization, as well as alter carbon and nitrogen cycling processes. And so with that, I'd like to take any questions. Thanks.
All right, thank you. We have time for one quick question. Any questions in the audience? Any questions online, Emily? No. Maybe I'll ask a quick question. You mentioned that this effect of fire on the cell microbiome is long-lasting within decades. Can you comment a little bit more how exactly you can establish that without, spending decades of time of waiting for the data?
Yeah, that's a great question. So I mean, we use a time for space substitution technique, which obviously can come with some caveats, and it holds the assumption that the forest that you are sampling as the undisturbed as the pre-fire condition, which of course we know is not the case. One thing I didn't speak about on this that compounds any sort of long-term ecological research is the impact of climate change. And so we know that the recovery 4 years after a fire in the 1970s is much different than 4 years after a fire in 2013 in terms of drought, in terms of heat, things like that. So it's a hard assumption to make, but the alternative is that we can't necessarily wait 40 years, 50 years to look at how these microbes succeed. So it's something that you need to sort of balance your conclusions with.
Makes sense. Thank you so much. Let's thank our speaker again. Thank you, Nick.